Good evening, everyone. Welcome. And welcome to the Barry N. Wish Professorship of Social Sciences inaugural lecture. As you've heard, some of you have heard me say at other lectures like this is actually one of the great moments in the life of a college where we celebrate extraordinary teaching and scholarship and the accomplishments of a colleague. So I could not be more thrilled to be with you tonight and to celebrate Rick Thompson. This professorship was created in 1999 by one of our trustee emeritus, Barry Wish, who was also a member of the class of 1963. And our speaker, honorary and honoree, and the holder of the chair tonight is Professor Rick Thompson. And he's only the second member of our faculty to hold the Barry N. Wish Professorship. The inaugural, or the initial holder of the chair, Professor Barbara held it from 1999 until her retirement in 1997, and Barbara is here with us tonight. Where are you, Barbara? There you are. It's 2017. What did I say? 1997. Oh, that would go backwards, so I messed that up completely. 99 to 2017. But we're thrilled you're here with us tonight, Barbara, so welcome. Rick Thompson is a most worthy recipient of this chair. As many of you have heard me talk about before, one of the really important challenges in front of us is continuing to evolve our curriculum into a more interdisciplinary and integrated curriculum. More and more we come to value finding ways that the different disciplines intersect with one another and engage with one another to find creative and thoughtful ways to solve problems is the way of the world. And Rick has been thinking in an interdisciplinary way from the beginning, since his days as an undergraduate at Pheromone, where he majored in both biology and psychology, and then his doctoral work at Cornell in biopsychology, and in his postdoc work in the zoology lab at the University of Oregon. And continuing the theme, and not surprisingly, here at Bowdoin, he is jointly appointed to both psychology and neuroscience. His research focuses on points where biology and social factors intersect, and specifically how molecules in the brain, including hormones and neuropeptides, influence social behavior, and my guess is we'll hear a little bit about that in a moment. Rick is a nationally renowned scholar in this area. He's been a principal investigator on 6 major grants from the National Science Foundation, the National Institutes of Health, and the National Institute for Mental Health. And he also recently took a leave here and served as director of the National Science Foundation's Neural Systems Program. His lab is not only a prodigious producer of important research and scholarship, but also of high-level student work. He has supervised over 50 senior honors theses and independent student projects. 21 of his students are first authors or co-authors on published papers, and 11 of his students have gone on to PhD programs in neuroscience or psychology. Rick is an exemplar of that which we value most highly here, great teaching and great scholarship. And I am very pleased to introduce him as the new Barry N. Wish Professor of Social Sciences. Rick, welcome, congratulations. Please join me in congratulating Rick.
Thank you, Clayton. I want to start by thanking Clayton, the administration, members of CAPT, and anybody else who was involved in the decision to name me the Barry Wish Professor of Social Sciences. Also my colleagues in psychology and neuroscience, some in biology, who have been so supportive of my research and teaching over the years, making this not only a stimulating place to work, but also a very enjoyable place to work. And I'd also like to thank friends of mine from the community who are not necessarily at Bowdoin, but who have really made this not only a great place to work, but a great place to live. And I know some of you. Thank you. Okay. So my work really focuses on chemicals produced in the brain and the body that influence normal patterns of social behavior. And I have 2 lines of research, one on neuropeptides that influence social behavior. And neuropeptides are simply short strands of amino acids, so like little proteins that are produced among other places by neurons in the brain where they're released and they act as central neuromodulators. The ones I study are vasopressin in humans and vasotocin in most other vertebrates, including zebra finches. Let's see if I can figure this out. Oops, I've just given away the punchline there. Let me make sure. Which one is it that is that? That's not it. Which button is the arrow? There, okay. So vasotocin, I'd get there eventually, you know. Vasotocin produced in zebra finches, rough-skinned newts, and goldfish, all of which I've studied in the last 20 years. However, I'm not going to tell you about any of that work today. So I apologize if that was a little bit misleading. That's the wrong button too. Going to get this right. Instead, I'm going to focus on my work on sex steroid hormones and how they influence behavior. This is work I've been doing more extensively lately. And so I'd rather sort of go into depth in that work rather than try to broadly cover the work on the neuropeptides. I'm going to try to keep this short. I only have 22 slides, though I have a few students in the audience I know I've seen. And as they can tell you, I can be prone to ramble sometimes. So I won't promise anything. And in fact, I'm going to diverge from the normative pattern of a typical academic talk early here. And actually, believe it or not, I'm kind of wondering, hard to believe myself, but I'm going to recite a poem that I actually did on my interview here 18 years ago at the request, I actually should say the demand of Barbara Held, who had seen that I had actually listed several poems I'd published on my curriculum vitae. So she therefore felt it was fair game and that I needed to be ready to say one. The content was appropriate then, and I think it remains so today. I'm going to figure this out eventually because I've already forgotten which one it is. Because it actually involves the hormonal regulation of courtship behavior in the species I'm showing up here, rough-skinned newts. So we'll see if I can pull this off. Tarica granulosa. A beaver pond like this can hold more newts than culture has answers for the questions of humans. It's my job to find them. The flash of orange bellies and another net full. Mostly males, slick-skinned and black-fingered beautiful this time of year. Their tails fat from testicular churnings and pulses of prolactin. The same inner voices that urge them to the edge, where they wait, like ripening high school boys beneath the window of some girl, likewise looking to explore. I've seen 10 tangled in a ball around a single female who went heavy on the pheromones before coming down to the pond to mate. Of course, these are not to be used as examples of courtship etiquette, unless to say, look, son, see how tightly that one wraps around her? As if right now nothing but the smell of her skin matters. So, it seemed appropriate. And speaking of testicular churnings, so what I'm going to be talking about are sex steroid hormones, and these are molecules that are produced primarily in the gonads, though not exclusively. They're all related from a common precursor. My gosh, you would think I would have figured this out by now. By a common precursor, cholesterol, through a series of enzymatic steps. And the sex steroids that I'm going to focus on today are going to be the androgens and the estrogens. And these are characterized that, well, the androgen that I'm going to focus on is testosterone, shown here. And it's essentially characterized by having 19 carbons in this common backbone structure. And the estrogen that I'm going to talk about, estrogens are characterized as having 18 carbons in their backbone structure. And the one I'm going to focus on is estradiol. So they're all biochemically related. And this is going to be an important enzymatic step that will come up again and again in the talk. So it's a very simple step to go from testosterone into estradiol, mediated by this enzyme aromatase. And this happens in a lot of cells within the brain. And counterintuitively, it's actually the conversion of testosterone into estradiol by cells in the brain that promotes a lot of male-typical social behaviors. But I'm not a biochemist, and the primary focus of my work is on social behavior and how these molecules actually affect normal patterns of social behavior. And if we're talking about the sex steroid hormones, we're primarily talking about how those molecules influence behaviors related to reproduction, be it courtship, sex itself, or aggression, which often shows up within reproductive contexts. And I want to point out, I'm going to get this. Let me see if I get this. I want to point out that I've italicized this word influence for a reason. These molecules don't cause any of these behaviors. Rather, they influence the probability that they're going to occur in particular social contexts, though the ramifications of those influences can actually be quite vast. One of the first of those really the primary reason that I'm interested in these molecules is because influences on how well an animal is able to attract a potential mate, potentially copulate once they come together, copulate once they come together, potentially form a pair bond, maybe care for offspring if necessary. All those things represent how these molecules essentially help animals solve what could be argued as one of the greatest challenges in nature, at least from an evolutionary perspective, which is passing genes on to the next generation. From a purely human standpoint, where psychological perspectives sometimes outweigh those evolutionary perspectives, some of our most intimate relationships are those that are associated with romance, sex, and parental behavior. So how these molecules influence those behaviors have a dramatic impact potentially on our day-to-day sense of well-being. And because those reproductive strategies, at least in social species like humans, operate within a group context, how these molecules affect those individual reproductive strategies have the potential to really shape how individuals within a group get along, and possibly even how groups interact with one another. Particularly if you think about the fact that intergroup interactions are sometimes, perhaps unfortunately, dictated by individuals who might be operating under such hormonal influences. Really? So it actually already popped up with what I thought was coming. The next question I want to address on this slide is why an interest in such a diverse array of species in terms of how these molecules work? And there are really two reasons for that. And the first is because I'm really interested in how the diversity in social behavior that we see in animals in the world around us gets produced. And sometimes that depends upon unique adaptations within steroid signaling mechanisms within the brain. So yes, I actually am really interested in how male newts are able to maintain this courtship clasp with the female for hours and hours and hours, which it turns out depends in part upon sex steroid hormones within the brain. Conversely, we sometimes see similarities in how these steroid hormone mechanisms work in a lot of different organisms from diverse vertebrate groups. And when we see those similarities, it really says that those mechanisms represent a general, you might say fundamental way that brain chemistry affects behavior. Something that evolved a long time ago, and that's actually been maintained throughout evolution in all these different organisms. And so when you see, and I should say too that the molecules that I'm going to be talking about, these sex steroid hormones, are actually structurally identical in all these different organisms. And the brains of these different animals are even more similar than you might expect. So sex steroid hormones actually act within the same parts of the brain in all of these different organisms, including humans, to influence these patterns of social behavior. So even if you're only interested in human behavior, when you see these similarities across all these different organisms and how these steroid molecules work, it starts to give you increased confidence that the same mechanisms might also be operative in our own brains, even if we can't study them directly sometimes. So ultimately, my work is about mechanism. That is, what are the nuts and bolts by which these molecules actually influence behavior? And we can start with the types of molecules they interact with inside of cells to induce those effects. And those molecules are what we call receptors. So these are the things inside of a cell that steroid hormones are going to stick to induce their behavioral effects. And there are two general types of receptor for sex steroid hormones. One of them is a receptor that sits inside of cells that mediates a slow effect that essentially slowly sculpts brain circuits. And let me walk you through this for a minute. So steroids are small lipophilic molecules, which essentially means they're greasy. They can diffuse through fatty membranes, and it turns out the outer layer of cells is a fatty bilayer. So these molecules, these steroids, can actually diffuse right through the lipid bilayer, interact with the receptor inside of the cell. These cells then translocate into the nucleus of the cell, where they essentially interact with the DNA, which is where the genes are. And so what these receptors are doing then is turning genes on and off. And that's affecting how much protein that these cells are making. This mechanism takes time. It takes time to produce sufficient changes in the amounts of protein within a cell to actually build brain circuits. So this, what I'll call genomic mechanism for the rest of the talk, often takes hours, if not days or weeks, to produce behavioral effects. Once those brain circuits are built, they then tend to stick around for a while. So a classic example of this genomic mechanism is actually sexual differentiation, whereby around birth in most animals, timing differs a little bit, species by species, elevations of sex steroid levels essentially build the circuits that promote or that make possible male and female typical patterns of social behavior throughout life. However, we also know that steroid hormones can bind directly to receptors that sit right on the surface of cells. And these receptors mediate a very different type of mechanism, and this is the one I'm going to focus on for the rest of the talk. So when the steroid sticks to these receptors, it actually induces very rapid changes inside of the cell that can immediately or very rapidly change the physiology of the cell. That can include altering the passage of charged particles into and out of the cell, which is essentially changes its electrical properties. And because neurons within the brain communicate through changes in electrical activity, this means that steroid hormones can very rapidly modulate the flow of information within the central nervous system. And so now you've got the possibility that steroids can play a very dynamic role in the regulation of behavior on a minute-to-minute basis. And this is where we're going to be focusing, is on these membrane types of mechanisms. So we know that this kind of mechanism that changes cell physiology very rapidly does influence behavior on a pretty rapid time scale. What we know very much less about is actually where within the nervous system these rapid membrane-mediated mechanisms are occurring. In theory, they could affect a number of different systems, and this is just a schematic of a goldfish brain. So you've got some eyes out here representing the peripheral nervous system, where sensory information is first perceived, sending that information to the brain. Some areas that I'll come back to throughout the talk are going to be the preoptic area, and this part of what we call the forebrain, and these big bulbs, which we call the optic tectum. We have the same parts of the brain as is shown here in the goldfish. And then in the back of the brain, the hindbrain. So we'll start out here at the front with these sensory processing mechanisms. It's possible that steroids can rapidly alter the way sensory systems process information, and therefore change the way an animal actually sees or hears, if it were in the ear, or smells, if in the nose, the world around it. It's also possible steroids could work within the central nervous system inside the brain. This is where that sensory information is coming, getting integrated into behavioral output. So sex steroids could also rapidly modulate this integration within the central nervous system, perhaps changing the motivation of an animal to actually approach or withdraw from another individual. Finally, to actually produce the behavior, there's got to be a motor output system somewhere, represented here in the hindbrain by descending neurons that go down to the spinal cord and eventually control the motors that are necessary to produce behavioral output. So it's also possible that steroid hormones can rapidly modulate this motor output. In fact, we think that it's probable that steroid hormones can in parallel rapidly alter all of these systems to prepare an animal to respond to whatever the social context is. And I'll just point out at the front end, we probably know the least about rapid effects on sensory systems. In fact, I'm going to argue that there's not any data out there yet that steroids can rapidly alter sensory systems. And there have been no examples yet where steroids can, in parallel, rapidly modulate multiple systems within the brain. So what I'm going to tell you about today is our work showing steroid effects, we think, rapid effects on sensory processing mechanisms, as well as what I think is going to be evidence for rapid effects across parallel effects across brain systems within a single species. The next question becomes when. When are these mechanisms actually active? What is the context in which sex steroid hormones produce these kinds of rapid effects? In the early 80s, a guy named John Wingfield went out onto the territory of male songbirds, and he essentially set up a net and then had to put a boombox somewhere underneath it and played the sounds or the songs of potential competitors that would simulate a territorial intrusion. He was able to rapidly catch the males that owned these territories, that swooped down to essentially meet this challenge, and show by rapidly collecting blood that this social challenge actually elevated levels of testosterone within 5 minutes of the social challenge. So essentially, he hypothesized or proposed that these rapid surges in sex steroid hormones probably help these animals mount an immediate response, in this case an aggressive response, that would help these animals essentially defend their territories. Since then, a number of us have tried to expand this basic challenge hypothesis to say, we now know that a number of different social stimuli can elevate levels of sex steroid hormones very rapidly, both in the periphery and in the brain, and that those rapid elevations of sex steroid hormones probably produce rapid effects on a number of behavioral outputs, particularly behavioral outputs that are going to in some way facilitate reproductive success, because a number of those social challenges do occur in reproductive contexts. And the idea here then is that those surges can probably rapidly amplify activation within a number of those brain circuits that I just showed you a moment ago, from sensory processing areas to central integration areas to motor output, to essentially allow the animal to respond in a maximum capacity with whatever behavior is going to be able to meet the social challenge. So to quote Nigel from This is Spinal Tap, the idea here is that these social challenges essentially elevate steroid levels that allow the system to go to 11 on demand. And I should never do an accent in a public talk, and if you've never seen This is Spinal Tap, you should. It's a good movie. So this brings us to the question of why look at these mechanisms in goldfish or carp? And I actually show you a picture of carp here, which is essentially the ancestral species of goldfish from which goldfish were derived through domestication. And I'm going to make the argument that essentially this species is an ideal one in which to understand how surges in steroid hormones might rapidly alter sensory systems and possibly rapidly alter parallel processes within the central nervous system. And I've shown you this picture of carp for a particular reason. They, like goldfish, essentially live in large shoals, social groups, and females ovulate across the spring breeding season, and when they do, multiple males will court and spawn with these females. So there's intense competition to mate with these females when they spawn. What's great about, and so goldfish have the same essential breeding patterns. And so what's great about this is it allows us to study how steroid hormones might rapidly modulate sensory and other behavioral processes that essentially allow these animals to adapt to this competitive context and display behaviors that are going to promote reproductive success. Because goldfish are a domesticated species, they're much easier to work with, yet we can understand those mechanisms within the context of the evolutionary pressures that shape those mechanisms to begin with, that are similar still with their ancestral carp species, ancestors. Okay, so what is the context for this rapid testosterone signaling that I'm going to talk about? So in goldfish and in carp, As in most other vertebrates, including humans, not only do aggressive stimuli elevate levels of testosterone, but so do sexual stimuli. So in goldfish, exposure to mating stimuli rapidly elevate testosterone levels in males. We therefore hypothesized that those testosterone surges might actually rapidly modulate behavioral responses that would facilitate reproductive success in those competitive mating contexts that I just showed you. A couple of corollaries to that hypothesis. First, we predicted that those mechanisms might be mediated by the conversion of testosterone into estradiol. And why do we think that? And it's to remind you of this enzymatic pathway here, whereby in many cells within the brain, that testosterone is actually converted into estradiol via that enzyme aromatase. So that's corollary number one. Corollary number two, because this aromatase is actually found not only throughout the brain, but also in a lot of peripheral sensory processing areas, that those mechanisms may be operative in central brain structures as well as in peripheral sensory areas. And so this is going to be a schematic that I'm going to show you often as we go through this talk, because essentially what we're going to do is start to test this pathway to see if this biochemical conversion to estradiol is important for rapid testosterone effects, if we can show that it's mediated by a rapid membrane mechanism, both within the central nervous system and the peripheral nervous system. And I should say that these rapid behavioral effects that are mediated by these membrane steroid receptors, by convention, and this is not absolute proof, but by convention, behavioral effects that show up within an hour of steroid manipulation are typically considered to be good candidates for being produced through these rapid membrane mechanisms. I'll also show you one other piece of evidence in addition to time course that we think more conclusively shows that. Okay, so we started with the visual system. And goldfish and carp essentially mate in the early morning as it's first getting light. And so the idea here was that rapid elevations or amplifications of visual processing mechanisms may essentially help these animals detect and or track potential mates in those competitive mating environments. I should say the other reason we had this hypothesis is because the aromatase that I mentioned is really high throughout the visual system of goldfish. So that includes out here in the retina, where there's a lot of aromatase, as well in areas where those retinal cells send their information, including the preoptic area, somewhere here in the forebrain, hopefully I'm close over here, and in the optic tectum. The optic tectum is an area that essentially helps animals orient towards whatever's moving out there in the environment around it. And the preoptic area is an area that organizes or integrates sensory information into behavioral output, motivated behavioral output, once the stimulus is perceived. And it was really the high levels of aromatase within the visual system that prompted this hypothesis in the first place. So we wanted to see, can testosterone rapidly enhance behavioral responses to the visual cues of females? And to do this, we, and by we I should say here I mean L.D. Lord, who was an undergraduate student working in the lab, essentially designed a paradigm by which he could measure rapid testosterone effects on behavioral responses to just the visual cues of females. So he set up a tank where he had a plexiglass barrier that prevented any olfactory communication between the animals. So a behavioral response here to the stimulus animal is just based upon visual cues. And he set up this tank so that essentially he could put the stimulus female in either side, measure the behavior of the male with these video tracking cameras, and essentially record the amount of time that this experimental fish spent in proximity, and that's usually within about 2 inches of the plexiglass barrier, to the stimulus fish. And typically what he would do is first measure the time they spend in that zone before the stimulus animal is added, then add the stimulus fish, measure the time at that point, and subtract that baseline score from the time when the stimulus fish is there to get the corrected proximity score that I'm going to show you in the subsequent graphs. So first he's just going to test this first basic idea. Does testosterone promote this approach response to this visual stimulus? And for this experiment, essentially he injected all the experimental animals with what we call vehicle on the first day. That's just the substance that he's going to dissolve all the hormones in for all of his experiments. So this is a control. And then for half the animals, he again injected them with vehicle on day 2. For the other half of the animals, on day 2, he actually injected them with testosterone. And this was a design that allowed him to use every fish as its own control. So if behavior simply changes as a function of repeated testing across the days, there would be a change in that vehicle-vehicle group. On the other hand, if behavioral only changes as a response of a rapid elevation of testosterone, the behavior should only go up on day 2 in fish that are injected with testosterone. And these injections, the behavioral tests, I should say, were done within an hour of the injection. So they were all done within an hour. The tests usually lasted 15 minutes. So here you can see the predictions, and in these graphs, essentially, I'm going to show you the 2 test days for each group along the x-axis. So here's day 1, day 2 for the vehicle-vehicle group. Here's day 1, day 2 for the vehicle testosterone group. And on the y-axis, it's just showing you the amount of time in seconds that the fish spent in that proximity zone close to the stimulus animal. And so what he found was that, as expected, if he just injected the fish with vehicle day 1 and vehicle again with day 2, there was no change across the days. And I should also say, if you're wondering what these are called error bars. They're just showing you the amount of variation within each group. So this is the average of all the fish on each day. And so essentially what we're looking for is variation between the 2 days that exceeds the variation within the days represented by these error bars. And for the vehicle-vehicle group, there's no change. So just as a function of repeated testing, the behavioral response doesn't change. However, when he looked at the fish that were injected with vehicle on day 1 and then testosterone on day 2, those fish began to spend significantly more time near the stimulus fish. And whenever I say significantly more time in this talk, we consider significance a way of testing if this variation between the groups is actually large enough to have not occurred by chance any more than 5 times out of 100, essentially. So this is just a convention within the field. And so whenever you see that something was statistically significant, you'll see little stars saying that this group spent more time near in the stimulus zone than this group. So this was consistent with this hypothesis that testosterone rapidly stimulates, at least within 1 hour, this visual approach response. This only happened if the stimulus animal was a female. So when he put a stimulus male behind the barrier, the testosterone no longer enhanced the response. So now, even though this group's injected with testosterone on day 2, they don't spend significantly more time near the stimulus male. As an aside, in case you're wondering, how do they tell the difference? We don't know. So it turns out that goldfish are not different within the visible spectrum. That is, we can look at males and females and we can't tell the difference. There's no clear coloration difference. It's possible that there's a difference in shape. Gravid females, that is, females that actually have eggs, have a more rounded body shape. However, the females we used were not ovulating, they weren't gravid, so we don't think that body shape made a difference. It's possible that the females we used displayed some kind of swim pattern that was attractive to the males. We did inject them with hormones that we know stimulate receptivity, so it's possible they were swimming in a pattern that was attractive to those males. I will say that the compartments we used were very small, which would have limited that ability. It's also possible there are differences in wavelengths that we can't see. So goldfish have receptors in the retina that are maximally sensitive to 365 nanometers of light, so light in the ultraviolet range. And when a student working in the lab, Emily Kim, doing an independent study project, also happened to be a photographer, essentially adapted a black and white camera with a lens that only let wavelengths less than 400 nanometers to pass. And then she took pictures of both males and females. And these are 2 males that she took pictures with and 2 females that she took pictures with. So this suggests, at least, that there are differences in ultraviolet reflectance patterns that allow these animals a simple way to differentiate male from female based on these different UV reflectances. But getting back to the testosterone story, so LD had shown that testosterone rapidly facilitates this approach response to the females. Now we wanted to know, is that mediated by this biochemical conversion into estradiol? So to do that, to test this, he did the following experiment. On day 1, he now injected every fish twice with vehicle. And that was so he could start to do some more creative manipulations on day 2. So for half the fish, he again injected half of them with vehicle first and vehicle again. So here's our control group, vehicle, vehicle, and then on day 2, everybody was injected vehicle, vehicle on day 1, and then on day 2, half of them were injected with vehicle first and then testosterone. So this should see if testosterone, even with these 2 injections, can still stimulate the behavior. And what you can see here, here's vehicle, vehicle, here's vehicle testosterone. So again, the testosterone is rapidly increasing the amount of time these fish spend near that visual stimulus. In the second half of the subjects, vehicle, vehicle on day 1, and then on day 2, the first injection was with a chemical called fadrazole. And what fadrazole does is it essentially blocks this aromatase activity, so it prevents the testosterone from getting converted into estradiol. And so the question was, would that prevent testosterone from inducing its effect on this visual approach response? And the answer was yes. So here's a fish, vehicle, vehicle. Now here's fadrazole first, and then testosterone, and the testosterone is no longer able to promote that visual approach response. Julia Bond, as part of her honors project, went on to show that in fact, if she just injected estradiol on day 2, just like testosterone, it stimulated this visual approach response. So together, these 2 studies essentially indicated that testosterone is promoting this visual approach response via this biochemical conversion into estradiol. We then wanted to know, it was a rapid effect within an hour, which is consistent with a membrane-mediated mechanism, but we wanted to see more conclusively, in fact, if that was a membrane mechanism, and also to see if it was being mediated within the central nervous system. So to do that, Julia Bond did a follow-up experiment where she used a different type of molecule. And so now the little E's that you see here are E2. These are the estradiol molecules, and they've now been conjugated to a large protein called bovine serum albumin. So essentially what this bovine serum albumin does is it prevents this estradiol from actually crossing the membrane of cells. So remember, steroids are lipophilic or greasy molecules that can diffuse right through the fatty layer outside of cells. This large protein prevents that from happening. She had to do, and the idea here is, if this molecule can still promote that approach response, then it would really be indicative that it's a membrane receptor sitting on the cell surface that's mediating that effect. She had to do a slight modification to the experiment because it turns out this protein would keep that estradiol from getting to the brain if we injected it peripherally, because large proteins can't cross the blood-brain barrier like small steroids can. So she had to use a surgical technique where she essentially, under anesthesia, implanted a small cannula into the 3rd ventricle of the fish brain. The fish wakes up. We can then go in days later and directly infuse chemicals straight into the brain, seal the cannula, and then test the behavioral effects of that infusion. She modified the design a little bit because these surgeries are very hard to do, so we couldn't use as many animals to get the 2 groups that I showed you before. So now she essentially used every fish in both conditions. Half of them got the vehicle, which is again just what this molecule is dissolved in on day 1, and then the estradiol BSA on day 2. The other half got the estradiol BSA on day 1 and the vehicle on day 2. So this allowed us to essentially control for possible order effects that might be going on. And when she did that, again, here's the visual approach score on the left, she found that the estradiol BSA directly infused to the brain did in fact increase the time these fish spent near the stimulus female relative to the time they spent near the stimulus female when they had been infused with the vehicle control. So this was consistent with the idea that this rapid effect of estradiol on this visual approach response is actually being mediated by a membrane receptor, and in this case that receptor is somewhere in the central nervous system since we were infusing this directly into the brain. So, central effect of estradiol mediating this visual approach response, but you'll remember I also said there's a lot of aromatase in the periphery, in particular within the retina of goldfish. And so now we wanted to ask whether or not estradiol might also rapidly affect sensory processes that may actually help males detect and possibly track the visual stimuli of females, particularly in the dim light mating conditions of early morning when they typically mate. So to do this, we had to develop a new technique. And the first thing we did to develop this technique was to recruit Patsy Dickinson to help us learn some electrophysiology so that we could actually record electrical responses from the cells at the back of the retina, as well as Dale Cyphers in physics. I don't think Dale's here to think about how to present naturalistic light stimuli. And so essentially what we do here is anesthetize the goldfish and then insert an electrode into the chamber of the eye that's essentially able to record the electrical activity of the cells on the retina. And these are the kinds of recordings we get when we do this. So we can flash a pulse of light onto the eye and essentially record what's called an electroretinogram. So this is not an action potential of individual cells in the retina. This is what we call a field potential. So it's essentially picking up on the combined electrical activity of the cells that are sitting back here in the retina. And what this shows you is that as we increase the intensity of the light pulse, we get a bigger and bigger positive deflection in this electroretinogram. And so this positive deflection is called the B wave. And because its size is proportionate to the intensity of the light coming in, it's often used as a measure of retina sensitivity. So in this experiment, what we were able to do is insert the cannula, insert the electrode, and then essentially we recorded a baseline round of recordings to this sweep of visual inputs at increasing intensity. We then injected the fish with vehicle, again what the steroid hormones are dissolved in, testosterone, or testosterone plus, and I'm not going to try to say the name of this molecule because I can't, so I'm just going to say PHTPP. And what this molecule is essentially something that sticks to a particular type of estrogen receptor called estrogen receptor beta. This is a receptor that's been shown to be in the membranes of cells, so that's why we started here. And once it sticks to that receptor, it prevents the estradiol from sticking to the receptor and activating it. So this PHTPP is what we would call a receptor antagonist. And so the idea here is that we're essentially, instead of injecting fadrazole, which blocks this biochemical conversion of testosterone to estradiol, we're skipping straight to the chase and trying to block the membrane receptor in the retina that we think may mediate those rapid effects. So we do our baseline round of recordings. We then inject the fish either with the vehicle, the testosterone, or the testosterone plus PHTPP, the idea being if testosterone is being converted to estradiol that normally activates this receptor, then this should block testosterone's effects. We then wait 15 to 20 minutes later, and then we do another round of recordings to the exact same stimulus input, calculate a % change from the first recording to the second recording, and compare across the groups. So that's what I'm going to show you in the next graph. And what we found was that at the lowest intensity, the testosterone treatment increased the magnitude of that B wave. So this is the change from the % change from baseline to the post-test score. And so here's 0, so no change. Here's the testosterone group. So the testosterone within 10 to 15 minutes elevated increased the magnitude of that B wave response, and it was statistically higher than the magnitude of the change for the vehicle control group. So here's your control group, and these are now medians, not averages. So here's your control group showing no change across the recordings. This was a significant increase in the testosterone group, and that % change was significantly higher for the testosterone group than for the vehicle group. That effect tended to be blocked by the ER beta antagonist. And I should say tended here to point something out. So the statistical testing I mentioned a minute ago, there's this magic number within science that essentially says that those effects, the differences between these groups, the size of this difference has to be large enough so as not to be accounted for by chance any more than 5 times out of 100. So this is probability right now. If it's smaller than 0.05, that is less than 5 times out of 100, that's what we call a real effect or a statistically significant effect. So we just missed that. So we had a P of 0.06. So this tended to block the effects of testosterone. Remember, this group had testosterone plus PHTPP. So the testosterone doesn't seem to be enhancing this effect, but we haven't shown that conclusively at this point. We'll show you some follow-up tests in just a moment, or I will. So at this point, what we can say is that testosterone rapidly amplifies the magnitude of this visual response to low-intensity light. So it seems to be increasing the sensitivity of the retina to light. What we then wanted to know was, does that general increase in sensitivity actually translate into increased retina responses to naturalistic stimuli, that is, to actual visual features of females? To do this, we had to change the way we do these tests. It would be very hard to use that same electrophysiological test to actually measure visual responses to naturalistic visual stimuli in awake, moving animals. So we developed what's called an immunohistochemical technique that I'm going to walk you through so you can essentially see how we do this. So we're again testing this part of the pathway, that there's a rapid membrane estrogen effect in the periphery, in the retina, that's potentially important for behavioral regulation. So to do this, we wanted to set up conditions as naturalistic or as close to naturalistic conditions as we could. And remember, goldfish mate in the early morning, so they've undergone sort of a dark adaptation overnight. Then they would typically mate as the light first starts to increase. So what we did is we essentially put males in the middle of a small compartment at night with lights out, and then we let them adapt in the dark. Early the next morning, Shauna Yu, who did this as part of her honors project, came in and injected the males, which were in this central compartment, with either, again, vehicle testosterone or testosterone plus the PHTPP. She then waited 45 minutes, and I'll explain why in just a moment, and then turned on the lights and added a female to all the compartments around the male. And the reason we put the male in the middle of this small central compartment with females all around it was we wanted to ensure that no matter what orientation this male took in this small compartment, there would be a female visual stimulus within the full visual field, and at a similar distance no matter where it was. So that if we observed any effects of testosterone on retina responses, it would be more likely an effect on sensory processing rather than a secondary consequence of that male simply looking more at the female or spending more time up close to her. So we turn on the lights, we add the females, and then we waited 90 minutes, and then we euthanized the animal, killed the animals, took out the retinas, fixed them, cut them in very thin slices, put them on a slide, and processed them for what we call phos immunohistochemistry. So what is that, and how does that allow us to measure retina sensitivity? So phos is essentially a protein product of a gene that gets turned on when cells become active. So seeing phos protein is essentially a proxy for seeing cells that were active. When they become active, they start making more of this protein. And we can that protein typically gets elevated, takes about 90 minutes for those elevations to show up in the cell after the stimulus onset. So our time course here is that by waiting this 90 minutes and then killing the animals and then looking for the phos protein in those cells, we're getting a snapshot of what was going on in the retina at this time point, which is 45 minutes after the steroid manipulations. So roughly a similar time at which we saw the behavioral effects of our testosterone administration in the earlier experiments. And the way we can actually see that phos protein is by taking advantage of the immune system, which can generate antibodies that selectively stick to particular proteins or molecules. Shown here is, I believe it says antigen on there. So we can essentially put an antibody on that will stick to the phos protein. So we essentially take our retinas, we put this antibody on, it sticks everywhere there's a phos protein, and then essentially we just take another antibody that likes to stick to the first antibody, and that second antibody has something fluorescent on it that we can actually visualize. So now we can see everywhere in the retina that there's going to be phos protein. And when we do that, it looks something like this. So here's the retina of a fish that just sat in the dark for this entire time. And you can see the blue is just background cells. So these are the nuclei of all the cells in the retina. And what you see is that there's no yellow or gold signal here. So this is the phos protein marked by these antibodies. And what you can see is that this is a fish that's now undergone this visual stimulation. So this fish has had the lights turned on and visual stimuli placed all around it. And you can now start to see this phosphoprotein showing up within several layers of the retina. So this is the internuclear layer, and this is the ganglia cell layer. So this is essentially a marker of cells that responded to that visual input. And our question is, does testosterone, through this estrogenic mechanism, increase the numbers of cells that are producing this phosphoprotein when they see the females? So this was Shauna Yu's work, and so essentially here's our control condition. These are fish that were injected with vehicle. These are the numbers of phos cells on the y-axis that we counted per, she didn't put the area down, but these are the numbers of phos cells. Here's our group that's injected with testosterone. So they had significantly more cells within this internuclear layer, at least, that we could see the phosphoprotein in than did the control group. So the testosterone increased the retina response to that visual input. Here's the group that was injected with both testosterone and the ER beta antagonist, and you can see in this case that ER beta antagonist did significantly inhibit the increase that testosterone otherwise would have caused. So the ER beta antagonist did block this testosterone effect in this case. A similar trend was observed in the ganglia cell layer, though it didn't reach significance, and I'd be more than happy to talk about that later. It's not trivial because these are the cells that actually relay that visual information to the rest of the brain. What I will say at this point is that what it appears, if we put those two experiments together, the electrophysiological experiment and the phos experiment, is that we get a general amplification of visual sensitivity as demonstrated with the electrophysiology that translates into an increased retina responsiveness to the visual input of this female. So you've got a general mechanism that's, we think, amplifying responses to whatever the most salient visual cue in the environment may be. In our experiments, that visual cue was a female that we placed around the males. In normal mating context, when testosterone would normally go up, that would likewise be potentially the visual cues of potential mates. But then things got complicated, or perhaps interesting. So this was work that a student from the University of Maine, Kimberly Deo, did. She actually wasn't starting off intending to look at behavior, but that's what got interesting. So she wanted to see what the effects of fadrazole by itself would be on actual courtship responses. And fadrazole, if you recall, is that chemical that blocks testosterone's conversion into estradiol. So it's this aromatase inhibitor. And here's what the behavior looks like. So there's a single female in here, and there are two males, and this is the courtship following that typically occurs. In this test, she actually infused a lot of pheromones into the water and also hormonally primed the stimulus female so that she would be releasing pheromones herself. Pheromones are simply chemical cues, in this case being released by the ovulatory females, that essentially elicit this courtship response in males. So this is a test paradigm that actually emphasizes, rather than visual cues, olfactory cues that drive this response. And surprisingly, what she saw was that here are fish that were injected with vehicle. Here's the amount of time they actually spent following that stimulus female. Here are fish that were injected with fadrazole, which is blocking the testosterone conversion into estradiol, and it actually increased the amount of time they spent courting and following the female. So what gives? Because if you recall in the visual experiments, when we blocked aromatase, blocked the creation or the production of estradiol, we actually inhibited the behavioral response to the female, and now we're actually seeing something that looks somewhat different. And what we think may be happening is that by blocking this conversion of testosterone into estradiol with that fadrazole, we know one of the things that happens is that you actually get increases in the amount of testosterone that those fish produce, as well as, and we haven't measured this yet so we don't know, but possibly another androgen that fish produce called ketotestosterone. So we started to wonder if what's happening here is that by blocking this biochemical pathway, we're elevating the amounts of androgens, and maybe the testosterone or another androgen is rapidly facilitating a response to an olfactory cue through a different type of receptor mechanism. This time we think a membrane androgen receptor. So these effects occurred again within under an hour, so a relatively rapid effect. So to test that, Megan Massa directly infused the ovulatory pheromones into the tank and tested whether or not testosterone could rapidly facilitate the time, or would rapidly facilitate the time that male fish would spend near the source of that pheromone. So now no visual cues involved in the test at all. So here you've got her control group injected with vehicle. This is the amount of time they spent in a quadrant in which she was infusing the pheromone. And here are fish injected with testosterone. Again, we're showing medians now instead of averages in these graphs. And in this experiment, the testosterone, again within an hour, significantly increased the amount of time that those fish spent in the quadrant where the pheromone was being infused. Christine Anderson, as part of her honors project, tried to repeat this test a little bit later in the breeding season, so the fish were actually no longer in full reproductive condition. She also not only repeated Megan's manipulation with just testosterone, but added a group, kind of like in our visual experiments, that had testosterone plus fadrazole. So the idea here was that in contrast to what happened in our visual experiments, where fadrazole blocked testosterone's ability to promote responses to the visual cues, we expected fadrazole would not block testosterone's effects on responses to the olfactory cues if within the olfactory system it's an androgen receptor and not an estrogen receptor. In this experiment, she actually didn't replicate the effect of testosterone. And so we think that as these fish transition out of the breeding season, they become less responsive either to the pheromone or to the testosterone itself. But what she did see was that the group injected with testosterone and fadrazole spent significantly more time near the source of the pheromone than did the control group. So this is at least, again, consistent with the idea that certainly it doesn't look like membrane estrogen receptors are facilitating this rapid olfactory response. So we need to repeat these experiments in full breeding condition, and that's what we're in the process of doing right now, to see if, in fact, there's a membrane androgen receptor that may rapidly facilitate this olfactory response. We're also starting to look and see if we can find that receptor in the brain. So for this experiment, we now injected into the brain testosterone conjugated to that bovine serum albumin. So you should remember this protein from when we conjugated it to estradiol. So now we've got testosterone conjugated to this protein, prevents it from crossing cell membranes. We inject that directly into the brain, only now this molecule actually has some fluorescent markers on it so that we can see exactly where it sticks within the brain. And when we looked, this is one of the areas where we actually saw what looks like a membrane version of testosterone. So this testosterone attached to the BSA can't cross these membranes, but it's sticking to cells. In this case, it's a part of the brain that's homologous to or the same as the amygdala in mammals. And so the amygdala in mammals is an area that we know, among other things, processes information, chemical cues used for social communication or pheromones. So we're seeing potential steroid binding, testosterone binding to the membranes within an area of the vertebrate brain that we know is important for mediating responses to social olfactory cues. This is just looking a little bit more closely at some of those cells. Actually, it may not be the same cells. This is, we now changed the technique, so we now are cutting the brain in thick slices, putting it right on a slide, and then adding this molecule straight to the slide. But what you can see is that these cells appear to have labeling all around the outside of the cells, and if we look even more closely, we can see not only is it around the outside of the cells, but it also seems to extend into some of the processes. So this looks like it might be testosterone binding to neuronal membranes inside of the brain. It's certainly not what it would look like if this were one of those receptors that I mentioned very early in the talk that mediates those slow genomic mechanisms, which are typically found inside the nucleus of the cell. So that would have been a little small dot right in the middle of that cell. So this is what our work is actively focusing on right now, is trying to figure out if there are these membrane androgen receptors within the olfactory system that rapidly modulate behavioral responses to olfactory social cues. So I said I would keep this short. I think I'm going to be good on my promise here. But I want to bring this back to the original idea that I put forward to begin with. And so what I'm going to argue here is that the mechanisms that we've started to identify in goldfish, whereby you've got testosterone being converted into estradiol, activating membrane receptors in both the central and peripheral nervous system, to rapidly modulate behavioral responses to social cues, as well as possibly activating membrane androgen receptors, perhaps in the central and/or peripheral nervous system, we don't know yet, but that these mechanisms represent a fundamental way that steroid hormones modulate a diversity of behaviors across vertebrates. And we think this because we're starting to see similar mechanisms in a number of species across vertebrate groups, suggesting this may really be something that truly is a general characteristic of how they produce effects on social behavior. Now, I'm not going to try to argue that these exact mechanisms are operative within the visual or the olfactory systems of any of these other species, but rather that variations on this theme, whereby a single molecule, in this case testosterone, can rapidly modulate behavioral responses through the activation of downstream membrane receptors within different parts of the central nervous system, possibly directly acting on those receptors itself, or being converted into other bioactive molecules that can themselves activate those kinds of receptors, can produce a diversity of effects on social behavior. What I'm really most interested in as far as the specific mechanisms we identified is the possibility that these sex steroid hormones can rapidly modulate peripheral components of the nervous system, specifically sensory processing areas, to actually change rapidly an animal's perception of the social environment, and central mechanisms that may rapidly alter its internal motivational states to approach or withdraw from other individuals. What's important to note is that none of these mechanisms mean that testosterone is going to promote a particular behavior all the time. Rather, that it may actually be able to promote context-dependent behavior. So think for a minute about the sensory mechanism that I just talked about. If you generally amplify sensory responsiveness, you may not only allow that animal to better detect a potential mate or a possible rival, but also to detect whatever else is going on in the environment around it, noting the context and perhaps recognizing that it's not the appropriate time to mate or to fight, and therefore promoting an alternative behavioral response. The possibility for flexible behavioral output becomes even more apparent if you think or realize that each of these separate pathways, each of these different circuits in which these receptors may be present, may each promote different types of behavioral responses, and that each of those circuits may be differentially regulated across social or cultural contexts, so that the same molecule testosterone may be able to promote different behavioral responses that depend upon whatever the social context may be. And if you think about all the different possible receptor mechanisms that are out there within these circuits, the sort of ability to generate diverse social outcomes becomes pretty limitless. I also want to point out that animals come different, even from the beginning. So individuals may produce different amounts of testosterone or different levels of enzymes within some of these circuits, different types or numbers of receptors within them. So you're going to have individual differences in how an animal may respond to testosterone. So not only may you get context-dependent effects of this molecule, whereby testosterone can promote one thing in one context and a different behavior in another, but you may have the possibility, too, where the same starting molecule testosterone can have very different effects in different individuals, depending upon how these circuits are built in those individuals, or possibly how past experiences have shaped these circuits in different individuals. So there's a lot of possible flexible behavioral outcomes that these kinds of mechanisms allow for in terms of social regulation, including individual differences too. Sometimes that variation may promote quite adaptive responses. It's also possible that extreme variation within these circuits may in some contexts promote dysfunctional social outcomes or be associated with difficulties that an individual may have fitting into a social context. So we think this is sort of the root, potentially, that underlies a lot of these diverse social outcomes. And I want to end the talk sort of stepping back and being a little bit philosophical for a moment. Trying to understand behavior from a scientific perspective, whether it be how biological mechanisms influence behavior or how social influences affect behavior, developmental experiences, or cognitive strategies, all come with challenges. It's hard to isolate the individual factors that influence behavior. And most of those factors that we try to isolate interact with a lot of other factors. And so it becomes very difficult to understand what that one thing is doing. And in fact, the amount of variation within a given behavior that's associated with a single factor that we're trying to isolate may in some cases be quite small. That can sometimes make it difficult to have the same certainty that when we manipulate that factor, that the behavioral outcomes that result from that manipulation are what we expected. These are real challenges. That said, I'll take those challenges every time when it comes to trying to understand behavior. That is, as complex as behavior is, and as difficult as it is to isolate those individual factors, I still would argue that behavior essentially operates according to the same natural laws that govern every particle in the universe. And that if we don't try to understand what those rules of life are, to sort of borrow a phrase from the Natural Science Foundation, we actually run the risk of starting to understand behavior in ways that at best are ignorant but benign, and at best that can be quite dangerous. So I really want to applaud people like Barry Wish, who have created a chair that's essentially dedicated to the scientific understanding of behavior, and to thank institutions like Bowdoin and others like it, who not only continue to support the scientific study of behavior, but also celebrate it on nights like this. With that, I also want to thank everybody who is involved in this work, primarily all the Bowdoin undergraduate students. These are all people who, in some form or fashion, made the experiments I discussed possible, as well as my collaborators, most notably Patsy Dickinson, who really made all the electrophysiological work possible, a former postdoc, Lisa Mangiamelli, past and current lab technicians as well. And of course, funding, the NSF, which now has a close place to my heart, and Embry for supporting a lot of the student research over the years. So with that, I will conclude, and thank you all for coming tonight, and I'd be more than happy to answer questions if you have them. And you're going to have to help me. It's hard for me to see hands in the back, so just sort of, I think there's, right there? I missed that if the test.
There's a receptor for the testosterone and estrogen.
Right. So why do you need to convert to estradiol? One possibility is that estrogen receptors do things that perhaps androgen receptors can't. And to get a locally high level of estrogen, you can't just elevate it in the periphery because it can be toxic, and there are sex differences in the levels of circulating steroid. So a way to kind of locally produce a specific effect of the estrogen would be to have this local conversion inside of those cells. Why evolution needed to do that, why it couldn't do everything with an androgen receptor, I don't know. One possibility would just be, if you want to build diversity, the best way to do that is by starting to change receptor signaling mechanisms. So that way you can start with the same starting molecule, in this case testosterone, and start to build all kinds of diversity by saying, in this circuit, we're going to work through an androgen receptor mechanism. And that circuit perhaps will use a membrane estrogen receptor. In another circuit, maybe we'll convert to a different bioactive molecule and use even a different receptor.
But what about the ratio of hormones? Have you tried to alter the ratio and see whether there's the actual ratio between the two?
So, in this case, that's always important, except what we're trying to do here is mimic what normally happens with regard to a testosterone surge. So we know that in natural context, there's not a change in the levels of estradiol. There's not even a change in ketotestosterone levels when males encounter female stimuli. It's just the testosterone that's going up. So what we're doing is just trying to mimic, here's what normally happens in nature. And so our injections are all creating elevations of testosterone that mimic what those natural surges are when males typically encounter females. In that case, are ratios important within local circuits? Maybe, but that's not matching what normally happens in natural context as far as what the peripheral levels of the steroid are doing. Does that, I mean, all I can say to that is, what normally happens is you've got a peripheral surge, and that's what we're manipulating, and it's producing behavioral effects that would be consistent with testosterone promoting these behavioral responses. What we're trying to do right now, too, is see if I've alluded to the possibility that all these mechanisms may ultimately enhance reproductive success. We're trying to set up some competitive mating paradigms to see if that actually translates into increased competitive responsiveness in those contexts. Why evolution did it, I have no idea. Other than, best answer I could say is it builds diversity. You know, why do pretty much every neurochemical system we know about have multiple receptor types that mediate those effects? Why go through all those steps? Yeah.
So with the last set of experiments you talked about, one of the things that caught my eye is you introduced a second male fish.
Uh-huh.
And I was wondering in the studies you did after that, was there always a second male fish there? And so for me, it's like, I know a lot about the testosterone, aggression, a second male fish.
Yeah, so in some ways, that experiment, per what I just said to Bruce, actually probably better mimics what happens in naturalistic conditions where you have multiple males that are going to be competing. We did that, again, because we were actually going into those experiments trying to test something very different, which is how pheromones affect the levels of milk production, which is essentially what the sperm is carried in, and we know that there are rapid steroid signaling mechanisms that promote sperm and milk production. So that was what we originally went in to test. It was just after the fact that we said, wow, the behavior really is changing in those fadrazole-injected fish in ways we didn't expect. This is, again, going back to wanting to set up competitive mating experiments, where it would be great to actually be able to manipulate one fish with an elevation of testosterone, one fish that doesn't get that, and see if that really does promote a competitive advantage to the fish that's got that elevation of testosterone. So moving more towards a naturalistic mating condition would be something that we're interested in doing right now. Yeah.
I want to thank you for a really superb Barry and Wish lecture. I thought it was complex yet accessible, and I really learned a lot from it. And I especially appreciate your reading your poem. Reciting it, I should say.
You know,
Yet again, but I didn't.
well, I figured it's fun to kind of come full circle on that. So,
You think?
yeah, I think
Glad
so.
I made the right decision on it.
Yeah, well,
But I do
thank
have a
you. Yeah,
question.
yep.
Of course, I mean, I thought the science was fascinating and your explication of it was very clear. And but I'm, of course, especially interested in your philosophical, self-described philosophical questions at the end of that, about all the challenges of the many variables that contribute to behavior.
Yep.
And the challenge, not only for any one, but for all in concert, as you indicate in looking at context-dependent behavior. You said that, you know, to not take up the challenge would be at best something benign, and at worst it would be something harmful. And it sounds to me like you have something in mind there, and if you could give some example of what you're talking about. I don't know if you're talking about the current political anti-science climate in this country or something else.
You know, I think I did mean it somewhat generally. I wasn't referring to the current political climate at all. But rather, you know, there are criticisms that I think we as behavioral scientists face in regard to replicability in part to the size of the effects we sometimes observe. And I think there can sometimes be a tendency to say, well, it's not a hard science, so why bother? The danger with that, I think, is that it's essentially punting, and it's essentially giving up on what I think is an important way to understand who we are as human beings, who animals are in the world, and what the reasons are for how we act. And if we start to abandon scientific ideas and understanding why we act the way we do, then we start to justify things, I think, that may be things that could lead to, I think, at worst, mistreatment of people. If we have certain biases or ideas about what meaning in the world and where that meaning comes from in terms of human beings and who's what and who's not. So it was really more a general statement to say, I really think it's important, despite the problems we have, that we continue to argue for approaching the study of behavior from that scientific basis, fighting through the difficulties that are out there, recognizing how important replicability is, trying to understand, why certain studies don't repeat, what contexts have changed, can we actually figure out why that's happening, rather than just knee-jerk sort of saying, well, yeah, I'll trust every kind of science except science that tries to understand behavior, which I think can happen sometimes. And I think that's a dangerous road to go down for very general, and I'll leave it at vague reasons for now. Okay, well, thank you all very much.